Okay, let's dive into a world that's completely hidden from us. A world where evolution doesn't take thousands of years. It can happen in just a few minutes. We're talking about microbial genetics. And trust me, it's a total game changer for medicine, disease, and how we even think about life itself. So to set the stage, let me hit you with a pretty staggering fact right off the bat. Around the world, bacteria and viruses are responsible for anywhere from a quarter to a full third of all human deaths. That is a massive, massive impact from organisms we can't even see. So, what's their secret? How can these tiny, simple organisms constantly outsmart our best defenses, like antibiotics? Well, it all boils down to two things. They are incredibly fast, and they love to share. So here's our game plan for this explainer. We're going to start with bacteria's secret weapon, this kind of microbial social network. Then we'll look at how they transfer genes directly through something called conjugation, and then cover a couple of indirect methods. After that, we'll talk about the packages of DNA they're sharing, the plasmids, and finally, we'll get into viruses, the ultimate cellular hijackers. All right, first up, let's get our heads around the main idea here. Try not to think of this as traditional evolution, the slow and steady kind. This is more like a super fast, underground social network, where the currency being traded is genetic code. You see, for most complex organisms like us, genes are passed down from parent to child. It's a vertical transfer. But bacteria, they've got a shortcut. They can pass useful bits of DNA, think of it like a software update for resisting an antibiotic, directly to their neighbors. It's called horizontal gene transfer, and it's what makes them so adaptable. And they do this gene swapping in three main ways. First, there's conjugation, which is basically a direct handshake. Then you have transformation, which is kind of like finding a useful thumb drive just lying on the street. And finally, transduction, where a virus acts like a little genetic delivery guy. Okay, let's zoom in on that first one, conjugation. This is the most direct method, the bacterial equivalent of a handshake, but instead of greetings, they're swapping DNA. Now, the key player in conjugation is something called the F factor. The F stands for fertility. It's this small circular piece of DNA, and it's absolutely essential because it contains all the genetic instructions for building a bridge to another cell to start the sharing process. This is a big one to remember for any exam. And this creates a really simple dynamic that you've got to know. A bacterium that has the F factor is called an F+ cell, and it's the donor. A bacterium that lacks it is an F- cell, and it's the recipient. Simple as that. But, it gets even cooler. Sometimes that little F factor doesn't just hang out on its own. It actually integrates itself, splices itself right into the bacterium's main chromosome. When this happens, the cell becomes what we call an HFR cell. That stands for high frequency of recombination. And you can just think of these as super donors, ready to share a huge chunk of their own genetic material. So what happens when that F factor decides to leave the chromosome? Well, if it's not a clean break, if the exit is a little messy, it can snag a few of the host's genes on its way out. This creates something new called an F' factor. And this is a really powerful tool for evolution because now it can transfer very specific bacterial traits to another cell. Okay, so that was the direct handshake, conjugation. Now let's talk about the other ways they get genetic upgrades, the more indirect methods of transformation and transduction. Transformation is, it's pretty much exactly what it sounds like. When a bacterium dies, its cell wall breaks down and its DNA can just spill out into the environment. And some bacteria, which we call competent cells, have this incredible ability to just absorb those free-floating fragments of DNA. The whole process is actually pretty straightforward, really just 4 key steps. First, the DNA fragment binds to the cell. Then, an enzyme chops it up into a more manageable size. Third, one of those strands gets pulled into the cell. And finally, that new DNA gets woven right into the host's own chromosome. And just like that, boom, the bacterium might have a brand new ability. And the third method, transduction, brings in that middleman I mentioned, a virus. To be specific, a type of virus called a bacteriophage, which is a virus that only infects bacteria. What happens is, during an infection, a piece of the bacterium's own DNA can get accidentally packaged into a new virus. That virus then basically acts as a courier, flying off and injecting that bacterial DNA into the next cell it finds. So we've covered how the information gets shared, but that begs the question, what is the information that's being shared? Well, very often, it's packaged in these little things called plasmids. think of plasmids as these little mini chromosomes. They're small, circular bits of DNA that float around separately from the main one, and they can replicate all on their own. And hey, that F factor we talked about earlier, yep, that's just one type of plasmid. And these plasmids carry all sorts of interesting genes, but one type is absolutely critical for us to understand, especially in medicine. It's the R plasmid, R for resistance. These are the plasmids that carry the genes for antibiotic resistance. The rapid sharing of these specific plasmids is exactly how we get dangerous superbugs. Okay, to wrap this all up, let's pivot and talk about the other huge player in this microscopic drama, viruses. They aren't just careers for bacteria, they're in a whole different league. And the thing about viruses that's kind of wild is that they're not really alive. They're basically just packages of genetic code, either DNA or RNA, wrapped up in a protein shell. They can't do anything on their own. They absolutely have to infect a living host cell to make more of themselves. And the diversity in their shapes is just mind-blowing. You've got everything from these simple rod-like viruses all the way to those bacteriophages that literally look like they came right out of a sci-fi movie, like a little lunar lander. And this variety lets them infect pretty much every type of living thing on the planet. And this really gets to the core difference. While bacteria have this sort of social network for sharing genes, sometimes for a mutual benefit, viruses are just straight-up hijackers. They inject their own genetic blueprint and turn that poor host cell into a factory that does nothing but build more viruses, usually destroying the cell in the process. Which brings us right back to where we started. Understanding this incredible, high-speed genetic network isn't just academic curiosity. It is vital. It's the engine that drives the evolution of all these pathogens. And it leaves us with a really critical question for the future of medicine. In this invisible arms race, with bacteria sharing resistance genes at lightning speed, are our strategies evolving fast enough to keep up?